Welcome to the Evidence and Reasons for the Christian Faith video channel. I'm Salvador Cordova. It is a pleasure to serve you this evening as I make my presentation on entitled Nerd Wars, Miracle Transitionals of Evolutionism versus the Stairway to Understanding Part 1. I actually don't know when Part 2 will come about. I'm still trying to get this idea fleshed out. This will be material that I'm thinking about for a future book. There are actually several booklets I'm considering working on. So just thought I'd let you all know that. I think a full-on book project is just too daunting a task, so I may not be doing that anytime soon. This channel is devoted to exploring a spectrum of evidences and reasons for the Christian faith. One verse that is close to my heart that you'll hear me quote often is that it is the glory of God to conceal a matter. It is the glory of kings to search out a matter. Many Christians and doubting Thomases rightly understand, I think rightly experience the feeling, and understandably, I'm not saying rightly, I'm sorry, check that. They understandably have doubts because they'll say, why, if God is so great, why isn't he as evident to our senses is the air we breathe? And it's only in the last few years I saw this verse. I mean, I've read it before, but it really stood out to me. And I said, oh my, it's the glory of God to conceal a matter. It is the glory of kings to search out a matter. It also says the kingdom of heaven is like a treasure hidden in a field. And when someone discovered this, the merchant discovered this. He sold all that he had. I'm not sure that it's a merchant, so apologies that I don't know this off the top of my head. But when a man discovered that there's a treasure in the field, he sold all he had and bought the field. And so there are a lot of things that God conceals to separate the sheep from the goats. God's children, God's elect, will seek him out. They will devote time and diligently search for him. I think that's one of the reasons that the Lord does this. Otherwise, we wouldn't even be in the business of apologetics, evidence, and reasons. There'd be no point in considering the questions at hand. So I have a section here in the YouTube website that is that covers my explanation for the hidden God and why he appears to some people and not others, and why he also conceals himself, as it suggests here in Proverbs 25:2. And in fact, I covered verses like where Jesus said, I praise thee, Father, Lord of heaven and earth, that you've hidden these things from the wise and learned and concealed them and revealed them to little children. And so I tried to give my explanation as from the scriptures why this has been the case. Let me try to greet everyone that has visited. And Cheryl, I saw you too late in the chat yesterday. You wished you had well wishes from my mother, and I didn't have a chance to acknowledge you. That's the problem when I'm running all the controls from my show, and I just can't get around to everything in the chat. So I'm just going to acknowledge everyone. Patrick, the Restream bot, Raman, Gavin, and Cheryl, thank you for joining us today in this presentation. So so that's about 4 minutes of preliminaries. And so this is going to be a topic close to my heart. The topic is the stairways to understanding. That's a single stairway, and this is I like this picture. It's a guy walking up from the earth, climbing a stairway to heaven. It's a very dramatic picture. And so the topic is this. This is a question that evolutionary biologist and paleontologist Stephen Jay Gould posed one time. He asked, did he, that is God, create to mimic evolution and test our faith thereby? Did he, God, create to mimic evolution and test our faith thereby? And let me explain why this is what this means. We're hearing that a lot of young students raised in Christian homes go off to college and they lose their faith. We know for a fact that evolutionary theory has been instrumental in a lot of people losing their faith. And I think that we underestimate, I think Christians dismiss the power of the argument. I'm not saying it's right, but something evil can be very, very powerful and deceptive. And we have to treat it seriously. So this is my goal. I'm going to explain kind of the evolutionary reasoning. And some people sometimes think I'm too friendly to the evolutionary biologist. And I'm just like, no, my purpose is to give proper respect to the power of your opponent. Because I have, this channel especially reaches out to biology students that are in there. They may feel outnumbered 100 to 1 in their schools by their classmates, and they have a professor who probably may be dissing creationism. So it's a different thing to be here on some YouTube channels where everyone's just laughing at evolutionists. It's different to be I've been in a hostile secular environment. And so what I try to convey here is what some people may experience in the classroom. It's not as easy as it looks. So again, and I was a little bit disturbed. People were actually questioning whether I'm a creationist. I'm a young earth creationist, a young cosmos creationist. I believe the universe was created less than 10,000 years ago. The exact figure, whether it's 6,000, 6,500, I'm not really particular about that. Also, too, if I could boast a little bit, there are a lot of people that hate my guts on the other side. Maybe I'll just point this out. I didn't mean to, but maybe I have some bragging rights here. There is this so-called Encyclopedia of the American Loons. And I'm just going to, let's see where it is. Yes, there's an Encyclopedia of the American Loons. And guess who's number 81? I'm actually listed as number 81 ahead of Sean Hannity. Here's what my opponents have to say of me. Here is another slimy, dishonest creationist and liar for Jesus. Cordova's style is well exemplified here. He is neither the most influential nor the stupidest of the creationists, but he's probably the most vile and dishonest. He has absolutely no scruples concerning quote mining, twisted misrepresentation, and blatant bald-faced lying to make a rhetorical point, which might at first seem convincing to the ignorant. So, just a little bit of bragging rights. I actually was listed at number 81 in the Encyclopedia of American Loons ahead of Sean Hannity. How that happened, I think it was just by coincidence, but I thought that was pretty funny. So why is it that some creationists might even think I'm not a creationist, or they question me? I'm not always friendly to every creationist argument. One of the ones that I got a lot of flack for was that I said the use of the second law of thermodynamics is not a good argument for creationism. That was put forward by the venerable Duane Gish himself. And I've criticized it, and I got a lot of flack for it. I'd simply ask simple questions like, okay, so someone is in the living state and the dead state. Give me the change in entropy. Give me a figure for delta S. And when they couldn't do that, boy, I said, maybe you don't have a good argument. I went off to grad school to study statistical mechanics and thermodynamics, and I realized it really is a bad argument. Maybe not quite so bad. So bad is what some of the incarnations of information theory arguments. I do believe in information. That's not the issue. But it's the way you apply it. I actually will use an information theory argument today, but we have to be careful with how we apply it. So some people have been really offended that I'll say things like It doesn't matter whether idea is science or not. It's more important whether it's true or not. Same for creationism. And so I just discourage people from getting into some of these disputes. So I could give a whole list of things because I've debated this for 21 years. And so I am slightly alarmed to hear that some people will question my dedication to this. It's been my professional job to find faults in some of our arguments and try to improve it. So with that, did he, that is God, create to mimic evolution and test our faith thereby? So what is the argument? Here's another one. This is from an ID proponent. He was a revered ID proponent about 20 years ago. He was an ex-creationist, became a backslidden Christian by his own admission, but then came back to the faith. But he believed in universal common ancestry, but he still believed in intelligent design. He's very much like Michael Behe in his views, And he asked this: if God wanted to persuade us of special creation, then why would he make chimps that look so similar to man? And I'm going to try to answer that question today, because for a lot of people, and myself included, it's like, Lord, I accept that you created, but I don't know why. Why'd you make a chimp? You're making the creationist argument harder because you made chimps. And in fact, it probably goes a little deeper than that. I'm sorry about that. In his image is saying that I'm keeping him up so late. So I'm sorry about that. So this is an older version of molecules to man evolution. It may not agree. It certainly doesn't agree with some of the other later versions, but this has probably had a lot of influence from Ernst Haeckel. So there'll be some terms there. I provided a link in the video description if you want to grab where I got this from. And I'm sorry for the video quality, but basically, let me see if I could do this. If you start here from kind of all the pre-biological chemicals and you go this way, you have protocells, prokaryotes, eukaryotes, Blastea, Gastrea, which are Haeckelian Haeckel terms. And then you go here, you get kind of these worms, and it becomes a fish. And the fish becomes more and more like like these tetrapods, and the tetrapod becomes like these mammal creatures, and the mammals become monkeys, and the monkey becomes man. Now, need to point out that whatever the exact sequence is, we can always take an arbitrary set of creatures, and many times we can just line it up in terms of similarity. So we always get this kind of progression. So this is what Stephen Gould said, what Stephen Gould was saying. Why did it mimic evolution? Because you have this almost progression of creatures, especially in what we call the extant record. We have creatures that, like today, we have we have bacteria, and we have single-celled eukaryotes, and then we have many creatures similar to this in the extant record. And then we also have like the fossil record, and depending on how you interpret it, they'll make a sequence. So we have this sequence, and that's very troubling even to a creationist like me. I knew 20 years ago this was a bothersome problem. And why am I speaking about it now? It's only in the last couple of years I began to see it, especially when I work at the NIH. And I'm going to try to give you the answer up front, and then I'm going to try to develop it. Oh, also, by the way, we're able to build these, what we call nested hierarchies, where we just take say a single protein. In this case, this protein is topoisomerase because I built this tree. And you take the topoisomerase amino acid sequences from all these creatures here, like say the human, the chimp, a dog, a rat, a mouse. Gallus gallus is a chicken, a zebrafish, a worm, fruit fly, this plant called Arabidopsis, the yeast, and then this from E. coli, which is a bacteria. So you take these sequences, you put them in a computer, and it makes a tree like this. And it just groups them together in terms of similarity. And it looks like a family tree. And it's like, well, God, why would you do that? So here is my answer. So in his image, just since you need to get your sleep, I'm going to give you the punchline in the next 5 minutes, and you don't have to hang around the rest of the time, and you could watch the rest of the recording. So did he, God, create to mimic evolution to test our faith thereby? I'll say, it doesn't matter. I'll say maybe yes and no. Maybe it does test your faith because God does conceal things, but it doesn't mimic evolution. His premise here, mimic evolution, it doesn't exactly mimic it. And that's the first attack. I'm going to show you that it doesn't mimic it in the subsequent parts, but there's still that progression. Okay, so there's still that progression here. So when they say there are no transitionals, it's like, well, you could say that between a eukaryote and man, there are these creatures that are kind of similar or intermediate. I'm sorry. So we could say between a prokaryote and man, we could say that there's a yeast out there that's at least one step on the way, and then there are creatures that are a little bit more similar, and that can be demonstrated with kind of things like that. That's not exactly right, and in his image, and there's some subtleties here. I'm not stating it quite right. I need to clean that up, but I hope you get the spirit of the argument. So when I said in the video description, it's not exactly accurate to say that there are no transitionals. I mean, clearly, a creature like a chimpanzee is closer to a human being than a fish. So you could kind of say that chimpanzee is sort of like a conceptual transitional. It's not a real physical transitional. It never was, and neither was an ancestor. Okay, evolutionary biologists will say it's not a transitional, that there was some ancestor that's undefined that's a transitional. But I hope you get my point here. I'm saying conceptually we could line up the creatures in a way that we can go from something very dissimilar to something more similar. That's really my basic point, and that can be a little unnerving. So why did God make other creatures? Creation would be obvious if there weren't other creatures on the planet, but just made humans, right? I mean, well, maybe not. I'm sure that there's some diehard atheists will say, there's a way that humans could just spring up. And also, at least there would be no problem of the appearance of evolutionary phylogenetic trees if there are no other creatures. We wouldn't end up having, this is what kind of like a phylogenetic tree here. We wouldn't be able to have these things, and we wouldn't be dealing with people like Erica and Dr. Dan, because they'll be confronting us and say, why did God make creatures so that they create these trees? Here is my answer. And it really hit home. It really hit home in 2018. Oh my goodness, it hit home. Just think about it. What if God only made humans and no other creatures? How would you learn biology? Just think about that. That means for us to understand, say, the organs of humans and how they work while they're alive, not just dead, we'd have to be dissecting human body parts. Does anyone want to volunteer themselves for a dissection? Now, of course, God could speak from the skies as he did to the prophets and apostles, but apparently he chose to be subtle and allow us to discover the truth through science. So yes, I will say this. God could have done business another way and just shouted the answers about human biology from the skies. He could have taught us and audibly speak from heaven and say, this is how the parts of your biology work. But I want you to think about that, because I began to think about that when I was at the NIH, and it really hit hard. It really, really hit hard. I was to make a presentation on neurons, a particular receptor on a neuron It was an experiment by Rosamond, and I'm embarrassed to say I've since forgotten what that receptor was. It's something about the tetrameric structure of some neural receptor. I still have no clue. I quickly forgot. But I was assigned a postdoctoral student to help me with my class presentation. So this was a neuroscience class. All the students were required to make a presentation on a famous experiment in neuroscience. And I was assigned this project. And so they had all these neural cells that they were studying. And the graduate student, not the graduate student, the postdoctoral student was talking to me, and he was explaining, well, yeah, this is really cool. We have this particular type of cell that we use. It's an immortalized cell line. And I said, what kind? He said, it's embryonic. And I hesitated. I don't remember whether I asked him, but when I got home, I looked this up immediately. It was an embryonic stem cell line that was taken from an aborted fetus. An embryonic stem cell line that was taken from an aborted fetus. And I looked up more details of it. You can buy these fetal body parts on the internet for about $250. You can get a kit of its cells. This poor innocent child that was sacrificed for reasons that we don't know, but now just thrown out there for medical research. And the whole time I was studying this, I was just so troubled. I was just incredibly troubled. I said, God, this didn't have to be. And I thought about the Old Testament, where the picture of Jesus Christ dying for our sins was pictured in animals being sacrificed for us so that we could live. And I realized, we ought to be really grateful that there are animals out there that can be sacrificed so that we can understand ourselves, and be cured. Medical research is moving forward because we're able to sacrifice creatures that are similar enough to us to move our understanding forward. From that day on, I thank God that he made creatures like us, close enough that we could study them all the way from prokaryotes all the way to chimpanzees, so that we might live and find medical cures and understand that we are fearfully and wonderfully made. We would not understand that we're fearfully and wonderfully made. Ironic as it sounds, the reason we understand that we're fearfully and wonderfully made is because of this progression. And this is why I call this, either the progression is because of universal common ancestry, or it's a stairway to understanding that we're fearfully and wonderfully made. A lot of the breakthroughs in our understanding of genetics has been because we have studied prokaryotic creatures. When Marshall Nierenberg at the NIH, he eventually won the Nobel Prize for figuring out the genetic code, he used prokaryotic creatures. That was the breakthrough. It would have been miserably difficult to have been trying to do genetic research. By the way, question, what is ESC? ESC is an embryonic stem cell, specifically usually from a human. So if you hear embryonic stem cell, it's probably human, unless otherwise specified. And just to complete that thought, I had just to set the record straight, I had no experimental work whatsoever with embryonic stem cells, but I did report it for my class. I got a huge ovation and a lot of accolades for my presentation, but it was very, very sad. I couldn't help but think, as people were giving me applause for my presentation on what these embryonic stem cells discovered, I said, this is such a terrible, wasted sacrifice. There's no reason we couldn't have used chimps or mice or dogs for the same thing. Why do we have to use humans for that? Why do we have to use humans for this? So my perception of this tree here, this progression here, is now changed. I said this provides a stairway to understanding humans, and I believe, see if I could find it. It's stairways to understanding other things. See if I have a picture. Yes. So, sorry for all the fast turning of pages. So we might have a stairway to understand ourselves, but we can also understand other creatures. So we have stairways. This is my best depiction of stairways, that we can understand other things. So we can understand other creatures and ourselves. And it's kind of a labyrinth of stairways. This is a bunch of stairways in India. And that's a bigger picture of it. So that's why I use stairways plural. I use this plural. So I got some flack earlier today and saying I didn't use so-and-so's answer. I didn't use so-and-so's answer. Well, I'm sorry. This show is about me giving what was on my heart and my answer. I'm not trying to not acknowledge the other answers other creationists have given. However, Matthew McLean was on a show recently on another channel, and he is a paleontologist, I believe, and he showed this nested hierarchy, and he said he didn't give an answer why it exists. I weighed in the chat and said, this is my answer. So, again, I'm not going to represent this as absolute truth, but I tell you what, I began to be grateful. I began to look at this no longer as a challenge to creationism, because it gave me the answer why. It's so that we can glorify God. You'll see that we're fearfully and wonderfully made, and appreciate. We know His divine, His eternal power and divine nature through the things that are made. These creatures here in this kind of stairway help us to understand biology and see how it's fearfully and wonderfully made. Okay, so I have more to my presentation. Emery, you can go to sleep now and just watch the recording. You got all the juicy stuff right now. So, oh boy. Ow. Emery doesn't mince words. Oh my goodness. Okay. The rest of this presentation will also say that there's a mistake in Gould's premise. He says it mimics, did God create to mimic evolution? I'll actually point out that evolution cannot even make these transitionals. It has to be the transitionals in the quote unquote transitionals. They have to be miraculously created. And even then, they're not really smooth transitionals. But I'll even just show, let's just assume for the sake of argument that the transitionals are real. We need miracles to make them happen. And I'll show some of that, because I'm going to be talking some serious molecular and cell biology and protein biology. It's going to get nerdy real fast. Okay, the title of this was Nerd Wars. So this is, if there are guys out there if there are people out there that, want to have a serious scientific discussion, and it'll be nerd versus nerd. It's like, let's do this. And I'm just going to share some quotes. Einstein got some of the ideas right. Sadly, I don't believe he was a Christian, but he said, the Lord God is subtle, but malicious He is not. Nature hides her secrets because of her essential loftiness, but not by means of ruse. And the way the Bible says it, it's the glory of God to conceal a matter. The glory of kings is to search out a matter. And not that I agree with the Message translation, but I just thought it was cute. God delights in concealing things. Scientists delight in discovering things. So let's move forward. So are there pieces of the puzzle of human biology that are spread around creatures? I will, as a caution and as a disclaimer, the following diagram has been contested by some evolutionary biologists, so I recognize that, but this diagram was published in Nature. It shows here, this is a Venn diagram, it shows here that the human genome has these 2,963 genes. Some of them are shared with the chicken and some of them with the mouse. What's really interesting is there are 48 genes that the humans share with the chicken that it doesn't share with the mouse. There's 73 genes that the human shares with the zebrafish that it doesn't share with the mouse or chicken. And there are all these sorts of pan-genome diagrams. So it's as if also, if this diagram's accurate, and notwithstanding that there be some complaints about it, but if it is accurate, then pieces of human biology are spread out to understand it. It's spread out across so many species. We need to understand our own biology. We should study other creatures. And this is what we call the process of studying model organisms. So we have these things called model organisms where we feel that they have some aspect of the way it works that is close enough to human biology that we could study it. We can do all sorts of like really cruel experiments on them. I almost never felt sorry for a rat, but if you saw some of the cruel experiments that are done in laboratories around the world on rats, you might actually feel sorry, shed a tear for them, because it was just gruesome. So what do we learn from other creatures? Well, from the roundworm, we learn things about our nervous system. From the squid, we learn also things about neuroscience, because it has a very large nerve cell, an axon, part of a nerve cell, the giant axon. It made it very easy to insert needles or whatever we needed to probe the nerve. We learned so much about human biology by studying the squid. We also learn a little bit about introns, part of the eukaryotic gene, by studying a plant called Arabidopsis thaliana. And my professor said this, and this is him quoting him: paradoxically, humans share many more introns with the plant Arabidopsis thaliana than with the fly or the nematode, which are animals. Now, we've heard a lot about the ENCODE project. We've learned a lot about ENCODE-like data, which is what we call the chromatin modifications, by studying yeast, baker's yeast. We learn powerful things about how our genome and epigenome work because we studied yeast. This is a gift from God that we can do this. We study bacteria to understand how our genetic system works, the process of replication and transcription and translation of DNA. Mice we use for all sorts of medical experiments. And we have the chimpanzee, which, ironically, I need to point this out, they have forbidden, certain medical quarters have forbidden medical research on chimpanzees because it's considered inhumane. But it's perfectly acceptable to abort fetuses and sell their body parts. Not a lot of complaint about that. I'm sorry to just have to call out our culture on what's going on, but I just don't think that's cool. I mean, I'm glad that we have compassion on animals. In the Old Testament, I would imagine it was pretty heart-wrenching to be sacrificing these beautiful lambs for your sins. And so I totally get that we don't want to sacrifice chimpanzees. I believe I had a discussion with a colleague. He said that's really too bad. We're not using chimps as model organisms because they're the closest to humans. They would be the best experimental subjects to be able to determine the efficacy of pharmaceuticals before we bring them to humans. So what we're doing now is we are jumping from mice, and then clinical trials on humans. The better thing would be to take a model organism like a chimp because it's so similar to us. And again, just to drive home the point, what's the alternative to using model organisms? Does anyone want to volunteer themselves for a dissection? So that is my answer. Why did God create this pattern of similarity and diversity? It is a stairway to understanding ourselves so that we could see that we're fearfully and wonderfully made as God told us. We also are able to decode the designs and understand them better and realize that there is genius at every level. We're able to do this because we have this variety of species, and it fulfills Romans. And I had a whole list of scriptures that I was going to share. So I shared Proverbs 25:2 and Romans 1:18 through 20. I didn't quote the whole thing. Psalm 139 through verse 14. We're fearfully and wonderfully made. Proverbs 25:2. Now there's Luke 12:27 through 31, and that's why that's where Jesus said, "Consider the lilies of the valley, how they grow. They neither toil nor spin. Yet I tell you, Solomon in all his glory was not arrayed as one of these." We know this. This has far more meaning in the 21st century than at any time in human history, because we've had this stairway. We've had these stairways to understanding. And Patrick here, yeah, why not just grow a ton of humans in the lab to test out drugs? That's the future we are headed into. Yeah, really sad, really sad. But you see, I want, this is the epiphany I had. When I looked now at all these creatures, I said, they're provided by God for our benefit, to help heal us and to understand us. For the longest time, it was like a thorn in my side. It's like, Sal, you got to explain those nested hierarchies. Now, I have no problem with it. I have no problem with it. Whether you're old Earth or young Earth, or whatever, these are a gift of God. These patterns of similarity and diversity. So, stairways to understanding. And one thing I'll point out, notice that the stairway moves up. You climb up the stairway, and we could say in this diagram, this kind of Haeckel view of diagram, Ernst Haeckel view of evolution, this diagram, we go from simple to complex. You know, we start off from these prebiotic chemicals, and they get increasingly complex over time. But what has been devastating, both confirmed experimentally, observationally, and theoretically, is that creatures devolve. There is no reason, there's every reason that we should not. This would be like climbing a stairway. The selection, maybe one way to say this, and something that Darwin got completely wrong, natural selection devolves. It does not evolve in the sense of accumulating complexity. So creationists might actually value natural selection when it's correctly stated. Natural selection prevents accumulation of complexity. Natural selection prevents evolution. Darwin said natural selection is the mechanism of evolution. Actually, natural selection is a mechanism against evolution. Natural selection is a mechanism against this sort of evolution, this upward evolution. We have so many levels of evidence to this effect. So some people will say, oh, look, we were able to evolve this. And I said, how much did you lose? So you evolved one good trait, you lost 10. How is that upward evolution? Yes. Cheryl, this is beautiful. Darwin's delusion. Oh. And Emery says, working on this project now. Okay, so, all right, this is going to be a fun project. Natural selection prevents evolution. Natural selection prevents evolution. Darwin should have titled his book, Prevention of Origin of Species by Natural Selection. So thank you. So now I'm going to get now into some technical details, but some things to think about. Some people say, is God being deceptive? God being deceptive? Is he trying to trip us up? Well, there is the opinion of some of my colleagues that he is making stumbling blocks, like in 2 Thessalonians 11 through 12, chapter 2, 11 through 12, that he sends people a strong delusion so that they may be damned, who love not the truth, but have pleasure in unrighteousness. That's not my view, but that is, the view of some, and I just need to acknowledge that. On the other hand, just because a superficial look at something may deceive us, does that mean God's being deceptive? I'll give you a very simple example. If you put a pencil in water, it looks bent. This is definitely an example of an optical illusion. Now, would we say God's messing with our minds because he makes the pencil look bent? And actually, Snell's law kind of explains why the optics will bend it. We're not going to complain. At least I don't know of any Christians that would say, oh, God's being deceptive because he makes the pencil look bent when it's not. By the way, this was an unsolved problem for thousands of years until Snell codified the answer, which is now known as Snell's law. Actually, some other people had probably figured the elements of this before him, but he got credit since he's the one who kind of pulled it all together and just for whatever reason got the credit. Probably a stronger issue is geocentrism. The whole thing about geocentrism versus heliocentrism. It used to be that most of the top scientists believed that the sun and the planets and the stars orbited the Earth. Perfectly understandable. I have to be careful when I say reasonable versus understandable, because reasonable implies that it's correct and it's not. But it's understandable how the early people may have thought that. Very understandable. And just as an aside, John Calvin used to say that people that didn't accept geocentrism were of the devil. It's like, that's why one of the reasons I don't like being able to Calvinist, even though I believe in election and predestination. I don't like that. That's one of the reasons, because I just don't want to be, that's just baggage I don't need. It's neither here nor there. So anyway, so this is comparing the geocentric theory with the heliocentric theory. The heliocentric theory is very subtle. There is a planet that orbits the Earth, or you can say it orbits. It's not exact because the moon's big, but we could say the moon orbits the Earth, but then the Earth orbits around the sun. But the Earth is also rotating. So this is a very motion. And so this whole thing of, is God being deceptive because he makes the geocentric? I'm like, no. When you get enough data points, you'll get the right picture of the world. Same for Snell's law. And what I'm going to say is same for this stairway to understanding. This is not a progression that confirms evolution. Far from it. It is a stairway to understanding that's been misinterpreted. This was created for our benefit. I'll summarize my points later. Ray, you missed the best part. So sorry to tell you that. I didn't announce this talk because I wasn't sure what my schedule was going to be. If I can be a little bit more organized, I might have a little bit more announcement time before I throw it. So that's partly on me. I'm sorry that you're heads up on this. So we can know from science, if we look at this Haeckel version of human evolution, and I acknowledge again, this is dated. James Tour did an excellent takedown of why these steps from like the prebiotic materials, the protocell, and the prokaryote gone. I'm going to argue today the problem with eukaryotic evolution. Arguments could be made for the other stages like eukaryotes and multicellularity of animals. There's probably going to be problems along the way in transition from like a fish to a land creature in terms of all the mechanical things. I'm not going to address this area here today, but if you could start chopping down the roots of this evolutionary progression tree at at the base. It's like, well, why don't you just accept the rest as being specially created? That's kind of my point. So, and then also, I mean, this is one way to depict the evolutionary progression. This is another way to depict it in terms of a family tree. And I'll show you how we'll maybe do the progression. So say we started with a human. What's the next closest? It might be a chimp. We might pick a mouse as the next. And granted, I skipped some of this, which is actually, yeah, okay. And we'll skip like, the chickens and go to the zebrafish. And I leave that problem for the evolutionary biologists, why this isn't just a straight ladder. And then we start with bacteria. So let's just say hypothetically, we climb this evolutionary stairway from bacteria to something that's yeast-like, to something worm-like, to fish-like, to mouse-like, to chimp-like, to human-like. So I'll cover the microevolution. This is the topoisomerase protein. You'll keep seeing the word top or top 2A. These are the what we might say, quote unquote, microevolutionary changes from the topoisomerase in the homolog in bacteria, the microevolutionary changes required to go from bacteria to human. But they're actually, I'm going to show the problems with this microevolution scenario. So this is really bad because I'm going to falsify both microevolution and microevolution at this stage. This is really bad. If microevolution and macroevolution have problems at these levels, it's really serious. I'm not saying there's no microevolution, but microevolution cannot explain some of the transitions here, and they're not even macro transitions. I'll explain what I mean. So this is a diagram that some of you have seen me cover before. We have the amino acid spelling here of a zinc finger 136 in humans. And you'll notice there are these non-random patterns here. Like everything in the things in red here, you'll see that that's a C, and things in purple here are H's. So just imagine that I just had this amino acid sequence without the coloring. You might not notice that, but there's this definite non-random pattern here. And then there are other classes of amino acids, such as the threonine and serines, that can be grouped together as aromatic groups, and phenylalanine and tyrosine. So, I'm sorry, these are the aromatic groups. Threonine and serine are not aromatic. Oh boy, Dr. Carter's going to ding me on that. So I just corrected myself. That was a mistake. This purple here is the aromatic group. But anyway, we can group them together, and you can see the non-random patterns. Each line identifies what we call a zinc finger, and I'm going to do a show on this. But I want you to see kind of the architecture. You see how these colors are laid out? It highlights the features. Now, what we can do is we could take the human zinc finger and say a zinc finger in a pig, and you could see that we have very similar patterns. And in this case, there's 50 to 60% sequence similarity depending on your scoring method. So we could take the zinc finger across a whole bunch of species, and we can make these phylogenetic trees. No problem. And as I said, the evolutionists will say, that's proof of common ancestry. I'm going to attack that shortly. I'm going to show that these phylogenetic reconstructions are not proof of common ancestry. It's circular reasoning to assume that they are. Oh my goodness. I did, just when you think someone's not listening, it's a professor of organic and biochemistry. He said, good catch, Sal. Yes, I need to know my groups better. Thank you, Dr. Carter. And we're privileged to have Dr. Carter listening. And like I said, just like with geocentrism and the bent pencil, we can look at these phylogenetic diagrams and conclude the wrong thing. It's understandable, but it's wrong. Now, I'll show another protein, collagen. It's 25% of the protein mass in our bodies, depending on what numbers you look at. And collagen's a real protein. You can find collagen in like Walgreens. They sell it as a beauty product. I'm not endorsing it, by the way, just for the sake of completeness. And this is the amino acid spelling of the collagen. And there's another non-random pattern here. Do you see it? I highlighted it in red. Those are glycines, every third position. And we can also build phylogenetic trees with that. So you could see that the humans have a very similar pattern to the zebrafish in their collagen 1. It's collagen 1A1 specifically. And there's about 75% similarity depending on your scoring technique. But the thing I'm going to, and you can build these phylogenetic trees, and the evolutionists will say, oh, that's proof of evolution. As I said, I'm going to criticize this. The first problem is it should be very obvious that it's going to be hard to construct a common ancestor of collagen and a zinc finger protein. And I've asked this. I said, so there's no universal common ancestry for major protein families. In fact, the zinc finger appears in vertebrates almost out of nowhere. It would involve a lot of horizontal gene splicing from other proteins. And it's just not, the bottom line is this won't progress by microevolution. This is evidence that microevolution cannot account for proteins. You would require macroevolutionary steps, and I'm trying to show just kind of visually what a macroevolutionary step would be in terms of major protein families. And I've talked to Dr. Dan, and I think I have a even a screenshot of a side chat I had with him. He was very generous. I asked him, do you think all proteins descended from a universal common ancestral form? And he said, no. So the evolutionists will say, they used to say, they don't anymore, but they used to say, oh, we still believe in universal common ancestry from a single organism, but the protein's independent origins. I'm just like, but that's actually been kind of dispensed with, universal common ancestry from a single cell. Emery and I had a show on that. And I think also with Mia and Otangelo, we actually just looked at peer-reviewed literature, and they're just saying, nah, they're thinking of population of organisms. But Dr. Dan kind of affirmed the point that I'm making here. I will let him speak for himself what he believes. Okay, I feel bad if I'm even in the slightest misrepresenting him. But I think, by and large, the evolutionary community has quietly acknowledged what we call in creationism the orchard model, separate created kinds for protein families. And you can see the problem right here. You can't go from here in microevolutionary steps and build something like this. And one of the reasons is your collagen will break apart and it's going to fail to be a collagen, or vice versa. Or same is true for many other major protein families. It's just like trying to evolve a spark plug into a tire. All the intermediate steps are not going to be very functional. You don't have something in between a tire and a spark plug for cars. We find that in the man-made world of machines. They're just, we might have some things that you can micro-evolve, but there's some things you're just not going to macro-evolve, like say like say you have a nut and a bolt. You're not going to evolve a nut into a bolt, okay? There are lots of problems. What happens in the man-made world in the idea that we don't incrementally evolve things happens also in the biological world. So not only, I'm hoping to show then that we have a problem with macroevolution for the major protein families. What I'm going to show, it's even worse because there's problems with microevolution of even the same protein. So just to emphasize the point, the idea is with proteins, we have certain architectures like collagen and zinc fingers. In the man-made world, we also have certain architectures like pianos and cars and blenders. There are variations within these major groups. That's kind of, just extend that to proteins. It's kind of the same way, right? You're not going to evolve a piano into a car or car into a blender. You can evolve a blender to another kind of blender. All right, so there are problems with macroevolution of major classes of proteins. So all these experiments that evolutionists cite as evidence of de novo proteins, it's like, okay, how big of a change are you talking about? It's certainly not a macroevolutionary change, and there are problems with that. Oh, by the way, I need to acknowledge Ray Giordano. He said, I'm so glad for taking biochemistry in college. I only took 2 quarters, but it allows me to have a fighting chance of understanding what folks are talking about. I seem to recall, Ray, that you said that had led to you, that helped you in your Christian walk, that you began to study these things, and it pointed you to the designer. If that's not correct, apologies in advance, but I seem to recall that, and that's why I wanted to highlight, because I want Christians to understand a little bit more about biochemistry and structural biology. So now back to this problem. I'm going to argue that the transition from prokaryote to eukaryote is going to be problematic even at the microevolutionary level. So just jumping from, like, say, this E. coli to something like a yeast is going to be problematic. And the reason is this sequence here. We call a nuclear localization sequence. And I'm going to go a little deeper into this nuclear localization sequence. Let me see. So where I'm headed, I'm going to actually, I did say in the video description, I'm going to cover some of my professional work. And this is why I saved the ugly stuff for the last, because it's so distasteful. It's really hard, even distasteful to me, okay? Because it is my job and it is tedious. But some of my professional work is done with the topoisomerase enzyme. And in 2019, we did publish on this. Had a poster session at the conference on experimental biology. And no, this is not an April Fool's joke just because it was published on April 1st, 2019, 2 years ago. So what I'm going to do, I want people to, maybe it'd be great if I could have the chance to share with you all. I'm going to stop my screen share here. Let me do a screen share stop. I'm going to try to put up a topoisomerase video. It's only 2 minutes long, and you can see what it does. First off, I hope it's apparent why this particular enzyme is going to be so difficult to evolve. So let me do the share here. Chrome tab, share audio, and topoisomerase. Okay, I'm going to just maximize it. And
Let's consider what happens as DNA unwinds during replication. As DNA unwinds, it acts like this rope when we pull apart its two strands. As you pull the strands apart, twisting tension builds up in the rest of the coiled portion. It is actually adding one twist to the remaining rope for each twist pulled out of it. At some point, you can't separate the strands anymore. The remaining rope is too tightly twisted. If you relax your tension on the rope, it will twist around itself in a supercoil. It is releasing tension. If you want to keep pulling the rope apart, you have to release the tension periodically, and one way to do this is to cut the rope and splice it back together. This problem has been best characterized in small circular DNAs. There are two methods of dealing with this problem in DNA. One cuts only one strand of the DNA double helix, and the other cuts both strands. Let's look at the first. Topoisomerase I enzymes cut a single strand of the double helix, pass the other strand through the cut, and reseal the break, relaxing the overwound molecule, which now has one fewer twist. Topoisomerase II enzymes do the same thing but with both strands of the double helix. Topoisomerase II cuts both strands of a double-stranded DNA and passes another double strand through the break and then reseals the break. So if a molecule of DNA is supercoiled, topoisomerase II can remove the supercoiling, 2 twists at a time, to yield a relaxed circle.
sorry, I was muted. Wasn't that amazing? Let me point something out. How does this incrementally evolve? If you build a topoisomerase that has the scissors and just going in there and chopping up the DNA and it doesn't stitch it back together, it's going to shred the genome. How does this happen in incremental steps? How can that evolve in incremental steps? Or what if you have a piece that stitches things back together and you don't have the thing that cuts it? Or what if you have something there that is not able to find the knots to untangle? Oh, that's the other thing. After you cut it, you have to pass things around in the right way after you cut it. So it's not just stitching it back together. You have to take the trouble to unwind it first before you reconnect it. It's not going to do any good to cut it and then reconnect it, and you don't unwind it. So there are 3 steps, and this is surprisingly done with a protein that's only about 1,500 amino acids. So there are problems evolving this in steps because without it, it dies. Okay, so this is okay. They may argue that this is an origin of life problem or very close to it. Some people say, well, we didn't need topoisomerase to begin with, but somewhere early in the cell's evolution, supposedly, you would need topoisomerase to untangle the DNA. How could this evolve by natural selection? Now remember, I'll call it Emery's axiom here. Natural selection prevents evolution. This is an example of how natural selection would prevent evolution because partial steps would be just creating a mess, especially just having a pair of scissors in your enzyme and it's just shredding your genome to pieces. Bad, very bad. Okay, so now let me try to get a little organized here. What next did I want to show? I wanted to show my I'll share my screen now. So we saw at least one topo video. If we have time, I'll show some more. And by the way, I have challenged evolutionists on this, and of course, they're not going to argue. I love arguing topoisomerase. I love arguing the other ones, or these abiogenesis advocates. I don't know. It's just like you tell me why this is highly probable, why this is going to emerge naturally. So this is the spelling of the topoisomerase here. It's about 1,500 amino acids. I'm embarrassed. I actually should know the exact number. So I'm going to look that up. One thing I need to point out, to make a functional topoisomerase, you actually need to have you need to make 2 copies. So the gene will express this amino acid, but it has to do it twice. So the red and the blue are identical. And these 2 things connect together to make a functioning complex. So this makes the problem even harder. You need 2 copies, and somehow these copies seem to know how to connect together and work together, because this did not evolve, most likely, if it evolved at all. Of course, that's why I don't believe it evolved. You don't have just one thing floating around and it does half the job. This is like having a pair of scissors and you only have one of the scissors, one part of the scissors, only half just doesn't work. I mean, it might act like a knife, but it doesn't make it a good pair of scissors necessarily. Anyway, so this is one way of looking at the. I colorize some of the aspects. This is one level of organization with the colors there. The biochemistry people know what the colors mean, is in terms of alpha helices and beta strands and turns. But I'm going to show this one. This is the more important diagram. This is, again, the very same sequence spelled out. And I'm going to point out this part here in the red. This is what they call a nuclear localization sequence. Just keep that in mind. In bacteria do not have this sequence. Bacteria do not have this nuclear localization sequence. So now I'm going to show the importance of this sequence here. So when we build a phylogenetic diagram, it'll say, oh, the human is similar enough to the bacteria, we can assume it evolved. I'm saying, well, not so fast. You would have to microevolve this, and you'd have to do it in 1 generation, because if you don't, the creature's dead. So let me show the problem here. of your biology savvy, we have prokaryotes, bacteria are prokaryotes, and then we have eukaryotes. Humans are eukaryotes. The prokaryotes don't have this nucleus. See, there's like a little membrane here. You can barely see it. It's very thin. It's just like a little envelope that kind of keeps all the contents inside and prevents things from going in and out. We don't have that in the bacteria. So the quote unquote, the topoisomerase homolog, which are called gyrase inside prokaryotes, can just freely float around in here, relatively speaking. The topoisomerase freely floats around in here. Once we have this nuclear envelope, first off, that's a really, how do you have a creature with no nuclear envelope evolve one? I mean, that's a big problem, but let's just assume for the sake of argument, now we have the nuclear envelope. What's going to happen to the poor topoisomerase? It has to be able to, the topoisomerase will come out of the, and I should have had another diagram for this. The topoisomerase, when the DNA is transcribed, the RNA goes out here into this area in the cytoplasm where it encounters the ribosome. The ribosome will translate it to a topoisomerase protein. That protein has to go back inside. And this is where the nuclear localization signal is important. And physically speaking, this sequence is somewhere here. This is another rendering of the topoisomerase protein. There are all sorts of ways we can draw this up. So just think of this. So in the bacteria, this sequence up here is the bacteria, the first line. This sequence is the human. Whether it's human or some other eukaryote, the issue is bacteria don't have this localization sequence that's here. And this is the localization sequence. So now I'm going to show, let me bring up another video. And thanks in advance for your forbearance as I get all these things in line. Okay, so, all right, nuclear export. Okay, so remember I showed you that nuclear localization sequence in the topoisomerase. If that's missing, this is you can get an idea what's going to happen. Apologies. Thank you. I blew it there, guys. Can you give me a second chance? I'm going to try to put the audio. Thank God for second chances. Try again. So this is nuclear import, share audio. All right, I'll try sound this time. Apologies. I think I got it right this time.
The nuclear pore complexes are the only channels through which small polar molecules, ions, and macromolecules can travel between the nucleus and the cytoplasm. By controlling this traffic, the nuclear pore complex plays a fundamental role in the physiology of all eukaryotic cells. Most proteins and RNAs are too large to move through the complex by passive diffusion and must move by an active process in which appropriate proteins and RNAs are recognized and selectively transported in a specific direction. Let's consider proteins imported from the cytoplasm to the nucleus. Only a subset of proteins can enter the nucleus, and they are allowed in by virtue of having specific amino acid sequences called nuclear localization signals. Nuclear localization signals typically consist of one or two short segments of basic amino acids. Nuclear localization signals are recognized by nuclear transport receptors called importins, which carry the cargo proteins into the nucleus. With its cargo attached, importin binds to specific nuclear pore proteins in the cytoplasmic filaments. By sequential binding to more interior nuclear pore proteins, the complex is translocated through the nuclear pore. At the nuclear side of the pore, the complex is disrupted by the binding of a protein called RAN to importin. RAN carries a molecule of GTP. This binding changes the conformation of importin, which then releases its cargo protein into the nucleus. The importin-RAN complex is then re-exported through the nuclear pore. A protein in the cytoplasm called RAN-GAP, for GTPase activating protein, stimulates RAN to hydrolyze its GTP to GDP, an action that triggers RAN to release importin back into the cytoplasm. RAN plays a key role in protein import and export. Note that while RAN is bound to GDP, it cannot disrupt the binding of importin to a cargo protein. In an action that prevents the depletion of RAN from the nucleus, the RAN GDP formed in the cytoplasm is transported back to the nucleus by its own import receptor, a protein called NTF2. In the nucleus, another protein called RAN-GEF, for guanine nucleotide exchange factor, stimulates RAN-GDP to release its GDP and pick up GTP. In this form, RAN-GTP can disrupt the binding of importin and its cargo, triggering the release of the cargo in the nucleus. By the actions of RAN-GEF, found only in the nucleus, and RAN-GAP, found only in the cytoplasm, a steep gradient of RAN-GTP and RAN-GDP is maintained across the nuclear membrane, with RAN-GTP inside the nucleus and RAN-GDP in the cytoplasm. Similar to imported proteins, proteins are targeted for export from the nucleus by specific amino acid sequences called nuclear export signals. Nuclear export signals are recognized by receptors within the nucleus called exportins through the nuclear pore complex to the cytoplasm. RAN-GTP promotes the formation of stable complexes between exportins and their cargo proteins. Note that this same form of RAN does the opposite for importins and their cargoes. The effect of RAN-GTP binding on exportins dictates the movement of proteins containing nuclear export signals from the nucleus to the cytoplasm. Following transport to the cytosolic side of the nuclear envelope, GTP hydrolysis and release of RAN-GDP leads to dissociation of the cargo protein, which is released into the cytoplasm. Exportins are then recycled through the nuclear pore complex for reuse.
I hope you all enjoyed that. The wonders that God has made. And so now I'm going to go back to, so I talked about nuclear localization signals. What happens if this nuclear localization signal isn't there in the topoisomerase when there are changes in the cell? When you get a nuclear membrane, then you have all this. It's going to be a disaster, dead instantly. And this is just a microevolutionary change. This isn't a big change, at least in terms of, just changing a few letters. There are problems in, not only do we need to have this localization sequence, there's a subtle problem here. It has to be in the right position for that important to find it. Because if it's inside here, it's not going to work. You can't just get this thing arbitrarily anywhere. It has to be in the right position. So it's not just the sequence, but it's also the position. And I just realized something. This is only in, this is in creatures like humans. You saw about the export signal. There's also the import. This is nuclear localization. That's kind of an import signal. There's also an export signal. The topoisomerase can go in and out depending on a certain context. And we haven't figured out why it does that and under what circumstances. But human topoisomerase is really, really special. Now you remember all the things that topoisomerase does. It's only spelled with this letter. This is like not even a few side chats in terms of the number of letters, and it can do all that. I've asked biochemists, can you build something like this from scratch if you didn't know, if you did not have a model to start with? They said, just like, no way we could build anything this complex and also this compact. But the point is, let me see if I have this in my slide. This whole thing with the microevolution, and I'm sorry to I didn't, in the next iteration of this slide, they're saying, microevolution can explain the changes in topoisomerase between creatures. I'm just like, not so fast. I just showed where this phylogenetic tree is meaningless. They could say, it's related, therefore it evolved. And I like that circular reasoning because it doesn't solve the problem that I just highlighted. When we put this stuff through these phylogenetic software tools that are going to generate these trees, they'll say, this is proof of evolution. I'm just like, no, not so fast. That's circular reasoning. Just because you can build a tree doesn't mean that it evolved naturally. You would need miracles. And I tried to explain here, you need a miracle even just for this. So just, that's my criticism of this nested hierarchy. That is my evidence. This is one evidence I put forward, that even if we assume universal common ancestry, which I don't, but let's just assume for the sake of argument, like some of my ID colleagues, like Dr. Michael Behe, that nominally accept universal common ancestry. He recognizes we need the equivalent of statistical miracles for universal common ancestry to work. In other words, evolution needs miracles to make it work, and evolutionists don't want to admit it. I just gave an example here why this phylogenetic tree is not a proof that this can happen naturally. I gave the evidence here, and no one has challenged me on that. I respect Dr. Dan, and he said, can you do some calculations? I could say, well, yeah, we can do some calculations. It's the probability of this getting the sequence in the right spot. It's pretty remote by random mutation. I mean, I'm not going to have to go through all that. You could see that it's going to be pretty hard. We don't know exactly how big of a change this, we might be able to sacrifice a few letters, but it's not going to be easy. And you only have one shot. And he said, well, if you don't factor natural selection in there, I'm not interested. I meant to say when he appeared on my channel, when I was having a discussion with Dr. Change Tan and Dr. Carter on this, I wanted to say, I just showed natural selection would select against this, if you think about it. So if you factor natural selection, to use Emery's principle here, natural selection prevents evolution. It's a negative factor. It's not a positive factor. So it's not going to help. If I added natural selection, it'd even be worse. So no one has challenged me on this. I don't expect them to. because they're always they'll always respond with this playbook of we can build a phylogenetic tree and that's proof enough for them. And I've just shown that that's not adequate. It is wrong thinking. You can't use phylogenetics to solve problems like that. So now I'm going to complete this and then I'll wrap up. It's been long and I want to thank everyone for coming. So now I'm going to talk a little bit about my professional work. So maybe I'm going to go back to what the theme of this was, since many have dropped in late. And that's really also on me because I didn't give any notice. And let me just stop sharing my screen here briefly because I don't want to get you disturbed by all of my clicking through my slideshow because they're like over 100 slides. So let me summarize what this all was about, and then I'll talk a little bit about my professional work as it relates to this. So this all began with, let me see if I can, this question by Stephen Gould. I'm sorry, this is the proverb. It's the glory of God to conceal a matter. It's the glory of kings to search out a matter. God could have explained all this to us through the skies, but he's chosen to be hidden and forces people to look and search him out and search for the things that he's done. That's one way he separates the sheep from the goats. That's what I have asserted. You don't have to accept that, but that's why I believe some of this is hidden. And people that come out here late at night to learn this, I thank God that he's put it on your heart to find what I hope I'm supplying some treasures. So the question is, the theme of this is the stairways to understanding versus evolutionary theory. Stephen Gould argued, he said, well, it looks like things have evolved. And he said, did he, that is God, create to mimic evolution to test our faith thereby? And what I tried to say is, the evolutionists will look at creatures and they can kind of group them like this into a sequence that looks like you can go from a sequence as simple to more complex. And you go from like this bacteria here all the way to a human. And for a lot of people, that's powerful evidence. That's enough that if they just stop there, they'd think things evolved. And I said, well, why would God make this pattern? Why would he make a chimp? Someone who's an ID proponent, he's like Michael Behe. He said, well, if God wanted to persuade us of special creation, then why would he make chimps that look so similar to man? And he's a Christian and he believes in ID, very, very much somewhat like Seigart and even more so like Michael Behe, but he doesn't embrace special creation. But I said what changed it for me was when I was studying at the NIH. Just ask yourself what would happen if we didn't have these other creatures. How would we understand biology? We would require everyone to be sacrificing their body parts to understand. Isn't it wonderful that creatures, innocent creatures, can die in our place so that we can be healed? The picture in the Old Testament, which is really a picture of Jesus Christ, the Lamb of God, dying for our sins. In the Old Testament, it was represented by animals dying so to atone for us. Amazingly, this is paralleled in the medical industry. Innocent creatures die so that we can understand our own biology and therefore find healing. That is the point. That is why, what I've argued, we have this sequence. We have all these creatures. We can learn about our biology. We can start with very simple creatures to understand translation and transcription of DNA. And then we have all the creatures in between. And I gave examples of some things we can study. We have all these creatures. We call them model organisms. We can study squids, for example, to understand our nervous system. We can study yeast to study our epigenome, et cetera, et cetera. And I gave like a 10, 15-minute talk on that part of it. So, I invite you all to rewatch that part if you're so inclined. And I did want to say this whole thing with the localization, and I don't know how I missed this in my graph. See if I can recapitulate it. If the importeins and the nuclear pore complexes change, there's going to be a problem in evolving. And I want to show this, and apologize for being a little bit disorganized here. So I said we can, James Tour did a great job of arguing against these early steps in this progression. And then I argued against the eukaryotic evolution. We could probably make good arguments for the others. And I said we might have some problem with the evolution of fish to tetrapods from a mechanistic standpoint. But this whole thing, if we find variation across species in their importein complexes, that means possibly that all the proteins had to be simultaneously reformatted. This is going to be a major problem for evolution. And I only realized it as I was making this slide, but we could almost probably knock out all these steps too regarding importein evolution. That is a subject of future research. We could really just crush this phylogenetic ladder here and say, you need miracles to make this work. So that's subject to some more future work, but this could be really devastating. And that's just this one piece alone. There are going to probably be other pieces. And this stuff with the nuclear pore complex is just brutally complex and difficult to understand. I mean, this is a description of what's going on, just a little description in terms of the chemistry. So I hope you're sufficiently have been wowed by what God has made. Praise the Lord. This shows his great wisdom and knowledge. And so I think I beat to death these phylogenetic trees are not good explanations. And it's potentially the nuclear pore complex itself in its evolution or lack thereof could knock out this portion of the evolutionary progression. I'll also point out some of my professional work on this. As I said, we use model organisms to help understand human biology, but there are other things that are just so totally cool. And I'm going to cover that. One thing is that as I said, the Darwin devolves. There's pressure for things to devolve downward, not upward. And Dr. Joe DeWeese, and I wrote a paper in the CRSQ, and we also did work in secular, but we got to tell the story in the Creationist journal. So we reported our research in the secular journal, but couldn't really tie all the loose ends. It's in the Creationist journals that we pointed out why we don't believe this could evolve. And I gave some of the reasons with like the topoisomerase. If it cuts and it doesn't reconnect, you shred the genome, et cetera, et cetera. We found out something very interesting. You could take human topoisomerases, the various versions of it, there are multiple versions of topoisomerase 2, put it in the yeast and the yeast will live. We have experimental evidence you can't do the reverse. It's very complex to explain. Just to reassure you, we didn't do the experiments on humans. We did it on mice, but that's close enough that we could say you can't take yeast topoisomerase and put it in humans, but you can take human topoisomerase and put it in yeast. The yeast will live. It's not going to work the other way around. That's a very interesting pattern. And I'm just throwing that out there because people have been trying to make predictions as to, how proteins, about the properties of proteins across species. And it could be that as you climb up the ladder of complexity, the proteins are a lot more sensitive to change. So you can find this at the CRS website, Creation Research Society website. And what else? So I'm going to just go briefly the papers. I hope I get through this in 5 minutes because this is just miserably difficult and I'm not going to go into it. Some of you may have missed that I announced that I was listed in the Encyclopedia of American Loons. I'm number 81. Ahead of Sean Hannity. I did that earlier, so I'm going to just delete that. And let me see what else. So I mentioned direct coupling analysis in the video description. Let me explain where this is going. Direct coupling analysis is you can take all the sequences across species. So I showed you the topoisomerase, and you can have it across species. You can put this in a computer, and it's going to predict a three-dimensional fold, which is pretty awesome that you can predict. Let me see if I can get the fold of the topo back. Come back to me, fold. So you see this fold here? Right now we have to do things like X-ray crystallography to figure out how this molecule is shaped. But what's really cool is we could take the sequences, say from a human and all these other creatures, put it in this direct coupling analysis machinery, and it can predict the fold. It can predict the three-dimensional fold. So I provided a link in the video description. Part of my professional work relates to this, not specifically that algorithm, but to the general idea you can figure out certain properties of proteins by comparing it across species. What is really spooky about this is you won't be able to do this if, like, say you take a class of animals, a class of creatures out of your data set. So if you take out, let's say, the plants, your fold prediction will start to fail. Or if you take out the animals, if you take out the single-celled eukaryotes, etc., it'll start to fail. Now, this is like saying, oh my goodness, did plants have to evolve so that we could have these 3D folds and that they would have exactly those changes? Why can't we just take bacteria or yeast and do this? You need the higher level creatures to be able to help you complete the fold. So even if you assume universal common ancestry, you would have to be supposing that this kind of mysterious second order code in biology just had to evolve for some reason, for no other reason to help us do scientific studies. So I've said, what's the reason for the patterns of similarity and diversity? It's to help us understand we're fearfully and wonderfully made and all the properties of biology. But then this is a bonus. And I'm saying this is staring you right in the face. I'm saying this to the evolutionary biologist. This is a pattern that's staring you out in the face that we can do fold predictions based on the patterns of similarity and diversity across creatures. And if you didn't have certain creatures, your fold prediction would fail. It's as if these creatures had to evolve so that you could do fold predictions. And so that is part. So there are other papers here that talk about this. This is one of them. I know some of the people involved in this whole research effort, and there are labs across the country that are getting involved in this, that we're able to do biology by doing cross-species comparison. Again, God has made all the creatures in biology to help us understand human biology and also the biology of other creatures. It's this integrated code. And this is where actually information theory is actually useful. And I'm almost done here because I saved the worst for last. How do you like that? You usually save the best for last. I saved the worst for last because this is just technically just dense. So we might take a particular position of a protein across species, and we could see that it's spelled differently. I don't know if you could see, like, it's a Y in this creature. In some of these creatures, another it's H. A Y is a tyrosine, H is histidine. And then in another column, you have these alanines, valines, L-leucine, I think. So when we have variation between these two positions, it also has information. This relates to my professional work, where we detect these covariations here, and we call it mutual information. We're able to identify important protein interactions based on this. So again, this is an example of how, back to Stephen Gould's question, why did God put all these creatures? It's like, why not? Because it's helping us do. Why did he put it? It's because he wanted us to understand the wonders of biology. We can understand biology because he's put all these other creatures. We can understand our own biology because of these other creatures. So that's that. And let me see, I just had I'm almost out. So I showed you can go to the wiki entry on direct coupling analysis. And in fact, this was done some years ago, and I'm having the privilege of doing a lot of what's in this paper by Durston, Chu, Wong, and Lee. This was a paper way back, I think, in 2012. We are now implementing this information algorithm to do protein structural prediction, and we would not be successful if God didn't make all these other creatures. And so this is miserable stuff with entropy and information theory. And like I said, I told you there is a use for it, but it's a real headache. So I saved the worst for last, and I don't think I have much more to torture you with as far as that goes. Yep, that's it. So with that, let me just look at if there's any interesting comments here. Evidence reasons. I'm jealous you're a loon. I'm proud to be a kook. And people are saying good night because it is late. And natural selection devolves. Yes. That was the theme of Michael Behe's book, Darwin Devolves. And everyone's saying good night. And I don't have much else to say. That is pretty much the heart of my presentation, because I wanted creationists to give answers to fellow Christians who may not be creationists as to why God made this pattern of nested hierarchies and similarity. And I hope that you all are able to use it. I hope it'll be an encouragement to others. It's certainly been a privilege to have this chance to be of service to you and share these ideas and things from my work. So with that, have a good evening, and I look forward to seeing you all again. Take care, and